Here we are. Welcome everyone to the Zangwill Club seminars of the last part of the last term of the academic year. We have with us Professor Lucina Uddin. Lucina or Lucina? We say Lucina?
Lucina, yeah.
Lucina. It's the Italian mind that makes you say it in a funny manner. I been following her for a while. She didn't know until I invite her to another seminar or discussion, and I've been meaning to have her in these settings for her to tell us whatever she wants for a whole hour about what she's excited about, and for us to discuss for another half an hour after that. I could say a lot of things about her, but she's going to do a mini introduction and little path career, which we usually want to have in the Zangwill twice a term as an example for a different career path that people can take. But I will say I'm always looking forward for her papers. I found her that she combines technical ability with clever theoretical framework in experiments. I think that's it for me. Welcome, Lucina.
Thank you, Tristan, for this very nice invitation and introduction. It's always a pleasure. And one day when we go back to hybrid in person, maybe I'll take a chance to come over to the UK in person. But for now, I'm happy to be able to share with folks some of our work on brain dynamics and flexible behaviors. But I will start with a brief introduction of myself because maybe not everyone knows. I was actually born in Bangladesh, which is this tiny country next to India, which only became a country in 1971. And actually my parents immigrated to the United States when I was too young to remember, so really less than a year old, and grew up in the United States, mostly in California, where I went to the University of California, Los Angeles to get my undergraduate degree in neuroscience with a minor in philosophy, which was actually helpful, I think, for a lot of times people don't realize how much of neuroscience is philosophy. So I went on and stayed there for my Ph.D., my graduate degree in the psychology department at UCLA, majoring in cognitive neuroscience there. And then moved on to my first postdoctoral a fellowship over at the New York University School of Medicine in the Department of Child and Adolescent Psychiatry, where I started. This was 2006, so this was the first time I became involved in the scary world of resting state fMRI, and everybody said, "You're throwing away your career, it's all noise, don't bother looking at these signals." So this was the beginning of my illustrious career, was everybody telling me I was doing the wrong thing. But I continued anyway from 2006 to 2008, working in the field of resting state fMRI and starting to look at neurodevelopmental disorders like autism and ADHD. And then I did another postdoc, which I think is becoming more and more common. So I started at Stanford University in 2008 and worked as a postdoc there for a number of years before getting a K award, a career development award, and becoming faculty there for a few years. But again, not in my own lab at that point. So then I forgot about this. So while I was at Stanford for 5 years, I took a detour and went back to Bangladesh. This was in 2010. So very long story short, there was a teaching opportunity at Asian University for Women, which is in Chittagong, Bangladesh, which is the city where I was born. So I thought, okay, I have to do this because Stanford was partnering with Asian University to send teachers over there to teach for one semester and send students from there back to Stanford to come for the summer and do a summer program. So I thought it was a thing I couldn't avoid. Essentially, when I saw the call for teachers to go back, I thought, that is the country where I was born, I have to go. So I did that in 2010 and took a little detour from research for a while. So that I highly recommend taking a detour from research for a while to really get your perspective back. Then I came back to Stanford and had an opportunity to start my own lab in the psychology department at University of Miami, which I joined in 2014. So there I direct the brain cognition laboratory, and we started doing the work I'll tell you about today. And I also started a cognitive and behavioral neuroscience division there and a graduate program. So we have courses in this area now. And since I've been there for 6 years, that was time for me to go on sabbatical. So then I came over in July, in the middle of the pandemic last year. We drove across the country to San Diego, where I'm currently staying. I'm currently a visiting scholar here, and not visiting anyone because of the pandemic. So we're still doing virtual things, but I am technically in San Diego and technically a visiting scholar there. But because I'm not doing anything in person on campus, I have a lot of time. And last year, my sister had a baby, and so I'm spending most of my days I'm a nanny now. So now I spend, I was telling Tristan, I wake up at 5 a.m., I do all of my meetings, and then I go and watch my niece from 11 a.m. to the afternoon whenever my sister comes home from work. So actually right now, I'm not much of a scientist at all. I'm just a daycare specialist. So that's actually been a whole different kind of education. And I also recommend doing things like that once in a while, just to take your mind off of the daily routines. And in fact, it's easier if you don't have a kid, but take care of someone else's and then come home and get a full nights of sleep. So that's been my past year here, spending time in California. Those are my sort of introductory roots, and I'm happy to talk more about that later. But what we actually study in the lab is flexibility. And for me, the interesting thing is looking at kids with autism, for example, and why they often want to do the same thing in the same way and keep certain routines. Like maybe they prefer to wear one pair of socks and they don't want to wear any other socks, or they prefer to take a particular route to go to school. And if you drive a different way, they don't like to go in a different routine. So, the problem with inflexible behaviors is something we see clinically. But also, I think the case of the pandemic has shown us just how much we all need to be flexible in our daily lives. So now we're giving talks sitting in our chairs instead of standing at a podium, but we're also teaching online and having happy hours on the computer. And this all requires a big change in our thinking about how activities should be structured. In fact, a great deal of the last year has made us all much more flexible than we ever thought we could be, perhaps. So we're actually very interested in the brain circuits that underlie flexible behaviors and how they can be affected in certain conditions, for example, in autism. And our lab takes an approach of trying to understand how brain networks develop across the lifespan from childhood through adolescence into adulthood, and how that underlies cognitive maturation, or the development of more sophisticated abilities across the lifespan. And then what are the implications of atypical developmental, brain developmental processes for cognitive maturation. So we study in parallel typical adults, neurotypically developing children, and clinical populations. And we use a multimodal neuroimaging approach. So that is combining task and resting state, functional magnetic resonance imaging, diffusion weighted imaging to look at structural connections of the brain, and things like effective connectivity modeling, so Granger causal analysis and other approaches to look at interactions between brain regions as they develop across the lifespan. And a lot of our work takes these ideas from foundational studies in network neuroscience and foundational theoretical frameworks. I like to point out especially this nice paper from Marcel Messelem in 1990. Even before the widespread use of neuroimaging to study the human brain, he was postulating, based on neuropsychological and lesion work, that cognition is subserved by interconnected neural networks, and that complex behavior is mapped at the level of multifocal neural systems rather than specific anatomical sites. The idea that there's this many-to-many mapping between brain regions and function is something that really predates neuroimaging and modern neuroimaging techniques. There's also been a lot of computational work by folks here. I'm just giving one example of a quote from Randy McIntosh, where he talks about the importance of understanding the neural context that a region operates in. Because, as he says here, the functional relevance of a given brain area depends on the status of other connected areas. So even if you're just looking at these gray nodes and their activation profiles, the context in which they're embedded really can affect the way that that particular set of nodes might function differently in scenario A versus B, for example. And also, we're very serious about this notion of dynamics, or how networks can change across time in the brain. And so here's a quote from Luis Pessoa, where he talks about how a network needs to be understood in terms of the interactions between multiple brain regions as they unfold temporally. So, and this has all been much easier to do in the last few years, because as everyone now actually believes, we can chart large-scale brain networks in the resting brain using these correlation and other techniques. And it turns out, of course, the brain is never idle. It's never just sitting there doing nothing. So even when you're in a resting, calm state of not engaging in any difficult cognitive task, your brain is spontaneously cycling through these coherent patterns that aren't random, but really are structured into networks that we see involved in cognition, memory, attention, and all the processes that we like to study. So these low frequency fluctuations are always present in the brain, and we can take advantage of that fact to look at the integrity of large-scale brain networks in typical and atypical developmental populations. This year, because of all this extra time due to COVID, I was able to write a couple reviews on flexible behaviors. So the initial one was an invited review looking at cognitive and behavioral flexibility across human and animal models, and clinical considerations from the many years of study in this area. And one of the most widely used tasks to look at flexible behaviors is this Wisconsin card sort task you see, where individuals are asked to sort cards according to one dimension, whether it's shape or color or size. And of course, frontal lobe patients were famously shown to be impaired in this, and in fact would perseverate on making one particular type of choice. This is one of the earlier methods that was developed for studying flexibility. And in the animals, you have these paradigms, which are usually called reversal learning, in which a stimulus outcome or stimulus response mapping is changed, then the animal must change its behavior to match the new mapping. And I think there's a lot that's been learned about the neural circuits that support these kinds of behaviors from that careful work in animal models. In humans, at least, we think that from neuroimaging that these processes involve frontoparietal, frontostriatal, and midcingulo-insular regions of the brain. And here's a more specific review for those interested in autism of what I think is the current status of the field, looking at neuroimaging studies that examine flexibility in autism spectrum disorder. So what we've been developing for the last few years is some ways of testing flexible choices in the scanner in a way that children can do and that children with autism can do. So we borrowed from the developmental psychology literature. So it used to be that you would get the children at a desk and put cards in front of them and ask them to choose two things that go together. And here, they might choose the second and the fourth card because they're both blue. And then you say, okay, choose two other things that go together. And they might say, well, the third and the fourth, they're both rabbits, and et cetera, et cetera. You can do this kind of flexible item selection task and make it easier or harder depending on how many cards there are, how many dimensions there are to choose from. But when we wanted to put this in the scanner, we adapted it in various ways. And we also created a control condition where we say, okay, just press the two buttons that are highlighted so that participants are sort of told exactly what to do and they just kind of follow along. So we first started with this task looking at neural responses in adults, because we just wanted to make sure that we were developing something that made sense in terms of the previous literature. So Dina Dajani in the lab created this paradigm and ran about 30 subjects between the ages of 18 and 40, so just typical adults here. She did a lot of work to make sure that the behavioral responses in a computerized version of the task outside of the scanner were similar to what we get from people doing it inside the scanner. And she looked at efficiency, reaction time, accuracy, combined metric to see how people improve across various trials of this task. But at the end of the day, there was, as we would expect, robust activation in frontoparietal regions, cerebellar, basal ganglia, and singulo-insular regions when you compare these flexibility minus control trials. So the task is doing what one would expect to be happening when you ask someone to flexibly choose between these different items. Now that we sort of expected, what we were then interested to look at a little bit further was you tease a part of the specific regions in terms of their roles in flexibility per se, because we know from, this is on the left, some automated meta-analysis using neuroscience. And we know that the components of executive function, which are related to cognitive flexibility, show very high levels of overlap in terms of the brain regions that are engaged. So shifting, which is the aspect of executive function that's thought to be most related to flexible behaviors, shows this characteristic pattern of activation across tasks. And updating, again, a working memory aspect is again showing some of those same brain regions. And inhibition, meta-analyses of studies of inhibition show some of the same regions again. So all of these regions are involved very broadly across executive functions. What we did on the study on the right was look at a effective connectivity modeling approach called group iterative multiple model estimation, which uses a structural equation modeling framework and takes a user-defined set of ROIs and looks at the relationships among them. And that this was developed by Katie Gates, who's a quantitative psychologist at the University of North Carolina. And so in collaboration with her, we did this effective connectivity modeling using the task data set that I just showed you, collected from the flexible item selection task, and found that it's really the inferior frontal junction, which is primarily involved in the flexible item selection process, and the other regions are sort of downstream activated as a result of completing this task. So, and there's a lot of individual variability in the network connectivity supporting this task. So it's not that everyone is activating the same regions in the same extent. So this was sort of the way we set up the task to study typically developing kids, and then of course bring that to children with autism. And so we have another line of work in the lab that looks at the development of different aspects of executive function and looks at how brain connectivity, both effective connectivity and structural connectivity, changes between childhood and adulthood. Some connections strengthen in the service of flexible behaviors. And we also do some work looking with age as a continuous variable between like 6 and 85, for example, how network interactions change across the lifespan. I won't actually talk about this stuff today, but in our flexible item selection task, we were scanning, 30 or so typically developing kids. And of course, they activate some of the same brain regions, but to a lesser extent than the adults when we asked them to do this task. Now, the trouble with this study was that we had done a registered report many years ago, which means we said, here's our hypotheses, here's how many subjects we'll collect, and here's what we're planned to do and publish. And then we started collecting data and everything was going along fine, collecting data from 7 to 12-year-old children, collecting data from 7 to 12-year-old children with autism. Everything was going along, but then March 2020 came and we were only about 2/3 of the way through the study. And that was the end of data collection for all of us and for the world. Also, another unfortunate thing happened in March 2020, which was that we switched our imaging facility. We switched our scanner from a GE 3 Tesla to a Siemens. So basically we got a whole new scanner and a pandemic. And so this project is a little bit indefinitely on hold. So we don't have enough data yet from the children with autism to do the comparison that we would have liked to do. And I'm not sure exactly how we're gonna deal with this for this registered report. But for the adults, at least, we think that cognitive flexibility involves coordination among brain regions that support executive function. And both adults and children seem to engage lateral, frontoparietal, and midcingulo-insular networks during flexible item selection. And task-modulated connectivity of the inferior frontal junction seems to be very important for this type of flexible behavior. So we might have some abbreviated data to present from the children with autism, so stay tuned. But this is like many of you, we've had some setbacks, and this is one of those casualties of the pandemic, unfortunately. So in other work in the lab, we just use a lot of the public data sets like the Human Connectome Project to help us understand flexible behaviors and brain dynamics supporting them in typical adults. And one thing that we've been using for a number of years now is this sliding window approach to functional connectivity, where instead of averaging across, let's say 6 or 10 minutes of fMRI data, or a 30-second window and look at how whole brain connections change across these short periods of time. And you can use things like approaches such as k-means clustering to say, well, there seems to be a few different states here and we can cluster them together. And then compute metrics such as the frequency of occurrence. That is, how frequently does this particular configuration occur? Or the dwell time, once you get into a particular brain state, how long does that state persist? Or state transitions, which is how often do you see switches between these functional connectivity profiles, switches between brain states. These are, we're using a tool here developed by Vince Calhoun's group. And what Jason Nomi and the lab was able to do is take a lot of data from the Human Connectome Project, which of course has an hour of resting state fMRI data for each subject. So really plenty of time to look at these dynamics as they unfold. And independent component analysis has been used to parcelate the brain into lots of regions here. Here is just data from a few hundred subjects. And the same participants have also, of course, performed many tasks outside of the scanner. So we have data on various cognitive processes here that a few of which we were most interested in. And what he found was that across these participants, their brains seemed to cycle through 5 states pretty consistently. And I ordered them here from most frequently occurring on the left to least frequently occurring on the right. And what's interesting is that the individuals who do better on working memory and cognitive flexibility tend to spend more time in the states that are more frequently occurring, the ones that are loose in terms of their whole brain connectivity patterns, and less time actually in the less frequent occurrences of states, 3 and 5, for example. So there's a trade-off between how much time spent in each of these states versus the performance outside of the scanner on these various neuropsych measures. So this suggests that the greater cognitive flexibility in the adult brain here is associated with the propensity to occupy the more frequently occurring configurations that are characterized by attenuated whole brain patterns of correlations and greater functional connectivity variability. And this is just to show us one way in which dynamic functional connectivity approaches can reveal relationships between these sort of individual differences in brain dynamics and how they support flexible cognition. Now, that was at the whole brain level where we were sort of agnostic to the particular role of any given brain region, but we have a lot of work contributing to this model of neurocognitive network dynamics, where we think that there's some special relationships between some of these large-scale brain networks that are ubiquitous in the literature. So, for example, we often, many labs see this idea that when the medial frontoparietal or default mode network activity goes up, then the lateral frontoparietal or the executive control network goes down. There's sort of a push-pull relationship there. And for many years, we've been using causal modeling approaches to show that the midcingulo-insular or the salience network here seems to be able to predict what's going to happen in these other 2 networks. So in the dorsal anterior insula in particular seems to be a driver of switching between these 2 other states. So one of our visiting scholars from Berlin, actually, Rosa Stemke, tested this midcingulo-insular salience network dynamics in a nice group of subjects that she had collected data from in a very interesting task setting that I'll show you on the next slide. So she had first collected some resting state fMRI data from all of her 100 participants, but then she also had them do a task outside of the scanner, this attention task, that during which the participants were undergoing eye tracking. So in the task, basically participants are asked to press a button when you see the letter pop up on the screen, and they're cued to look over to where the letter will pop up. And sometimes the letter E will come on the screen, sometimes the letter F. And so the eye tracking will tell you, okay, they're doing what they're supposed to be doing. But then sometimes an erotic distractor comes up and gets them off target completely. And the eye tracking tells you, well, they're not paying attention where they should be. And so sometimes they miss the target. And you can get all this quantification of where their eyes are moving while they do this task. So that was very interesting. And as you can imagine, reaction times are impaired and error rates are higher when participants are viewing some kind of interesting distractor, and they sometimes miss the task altogether. And so there's a lot you can quantify here about the gaze distance and all of that. And as you can imagine, there's a great and how well people adhere to the task in the face of this distractor. So what she did was look at these insular and cingulate regions of the brain and their dynamics in terms of their connectivity with the rest of the brain in the resting state. And there was one state in particular, which here I'm calling salient state 4, where you can see there's basically a negative correlation or very low levels of correlation between the salience and the visual networks. And it turns out that those individuals who show more occurrences of that state are the ones who are the most able to resist the tempting distractors. So if you have a brain that's wired so that the visual network is less coupled to the salience network here, it actually makes you better at doing this task of resisting the erotic distractor. So this is a example of how we think, and interestingly, we didn't see the same kind of result for the lateral frontoparietal or executive networks. We only saw this for the midcingulo-insular. So we think that these network dynamics correlate with the capacity to resist tempting distractors. And this is just also to highlight how brain network dynamics may be related to individual differences in self-control. Now, when we talk about the insula, of course, it's ubiquitous in terms of you see insular activation across so many different types of paradigms. And here, if you look at studies, of course, in the affective domain, emotion, empathy, and pain are very much linked to insular activation. Subjective awareness, interoception, somatosensory processes, of course, also involve the insula. Then at the higher level, you have conflict, switching, attention, and inhibition. And again, you see insular activation for those types of processes. So it's that brain region that does everything in some ways. So what a number of us have been working on over the years is parcellation and understanding subdivisions of the insula. One way to do it is to look at the whole region and take each voxel and ask which voxels have similar patterns of whole brain functional connectivity, and then cluster together those voxels and say, well, that may be a subdivision of the insula. So these studies have suggested there's anywhere between 2 and 13 or so subdivisions within here, but I'm showing 2 examples from studies by Ben Dean and Luke Chang, where they suggest there's evidence for at least a dorsal anterior subdivision, a ventral anterior, and a posterior based on whole brain resting state functional connectivity patterns. If you look again at the meta-analysis on the left from Luke Chang, he found that when the ventral anterior insula is active, it's usually studies that involve the terms in red, like anxiety, emotion, etc., like the affective kind of paradigms. And what we did on the right is a meta-analytic connectivity modeling, another type of approach for looking basically when one region is active, what are the other regions of the brain that tend to be co-active with it? So here the ventral anterior insula seems to be co-active with many of the limbic regions that you can see on the right. The dorsal anterior insula actually seems to be more active across these high-level error, inhibition, switching types of processes. And if you look on the right, seems to also co-activate with areas of frontoparietal cortex. Whereas the posterior insula seems to be the one more engaged in pain, somatosensory, et cetera, interoception, and seems to be more co-activated with primary sensory motor cortices, as you can see on the right. So perhaps there is some subdivisional structure that explains some of these ubiquitous insular findings. But at the same time, all of these regions are active across all of the different cognitive paradigms. There's this common functional fingerprint that shows that across all task domains, there still is some level of insular activation of all of these regions. So there's a great deal of convergence as well. So one thing we thought might be helping to explain this is the dynamics of the insula. And if you look at the subdivisions again, here we did a sliding window connectivity analyses. And the long story short is that in not every state of connectivity looks the same. So even though in state 3, the different insular subdivisions show kind of similar patterns of whole brain connectivity with other parts of the brain, in some cases like state 2, there's widely divergent patterns where the different subdivisions are talking differently to parts of the brain. And the real sort of interesting thing to note is that the dorsal anterior insula is the one that's the most variable in terms of its functional connection partners. So dorsal anterior insula is different from state to state in terms of who it talks to, whereas the other subdivisions don't do so much of that. crosstalk. Paralleled by some studies that we did on structural connectivity of the insula. In fact, the dorsal anterior insula showed some unique patterns of fibers that we found going to frontal and subcortical structures that we didn't see for the other subdivisions that we looked at. So this suggests that in adults, the insular cortex can at least be parcellated into a dorsal, a ventral, and a posterior subdivision, with the dorsal more likely to be involved in cognitive control, the ventral more involved in emotion, and the posterior more involved in somatosensory processes. And finally, I'm just checking my time to make sure I'm okay on time, but I can't get my phone to tell me what time it is. Okay, and the dorsal anterior insula seems to exhibit variable functional connections with also unique structural connections that we think enable functional flexibility of this region. And also in our models of neurocognitive network dynamics, it tends to be the dorsal anterior insula that shows this driver effect that it turns out in a Granger causal sense can be used to predict what's happening in other large scale brain networks and not the other subdivisions for this particular function. And one thing we're hoping to do in the future is take advantage of intracranial recording to get a better precise measure of the timing of activity, which of course is hard to do with fMRI. But a lot of groups have already started using ECOG data at the surface to do the kinds of network studies and to confirm sometimes the findings from the fMRI literature. So I'm right now collaborating with a group at Western University in London, Ontario, where their neurosurgical unit in the epilepsy ward, they routinely plant electrodes in 3 different parts of the insula, the anterior, mid, and posterior. So I was very excited when I heard that they were doing this because I thought this would finally give us a chance to get more fine-grained information about insular differentiation and responsivity and the timing of the activation in these areas across different paradigms. So that was another project that was put on hold due to COVID, but it's just now resuming again. So I'm hoping we'll have some data to share in the near future. So this all gets us to the point of talking about clinical populations. And this is sort of the goal of all the other studies is to point us to what we should be looking at in disorders that involve flexible behaviors. And I think autism is very interesting because for many years, the focus has been on social communication deficits and and that's been the focus of a lot of attention and therapy and also of the neuroimaging literature. But there's some of the aspects of transitioning to adulthood and autism, which I think warrant a closer look. And one of them is that very few individuals diagnosed with autism end up living independently. And that's a shockingly low number compared to other disabilities, as you can see in this chart. And there's a similar grim statistic when it comes to employment. So you have a lot of unemployment with adults on the spectrum. Even with high IQ, you see this. And that's very low compared to the other disabilities that are shown here. And I think the social communication deficits are surely involved in contributing to this. But there's also flexibility and executive function deficits in autism that are often overlooked or not as well characterized. And I think when it comes to living independently and holding a job, the flexibility does become one of those critical factors, especially in COVID times. But when you think about taking a bus to work and then you hear that people are on strike and you can't take the bus. I think last time I was in Spain, there was this labor strike and I couldn't take the bus from the airport to the hotel and I had to figure out what other system I could take. And that's just a flexible behavior that we all engage in whenever we need to make a split-second decision on something. But having trouble with that kind of decision making and that kind of flexible behavior, of course, would be a setback in terms of independent living and employment. The good news is there's a few therapies that seem to work well for training flexible behaviors. One of them here, Unstuck and On Target, is an evidence-based one. And we don't really know exactly which kids will benefit the most from these, but I think that's something which we would want to find out. And one course, the heterogeneity in autism and other neurodevelopmental disorders. So not every kid with autism is going to show the same profile of executive function deficits as we and others have shown. This is a study where we used latent profile analysis across parent report, behavior rating, inventory of executive function measures. And we had a mixed data set, which included kids with autism, kids with ADHD, kids with autism and ADHD, so comorbid, and also typically developing kids in the 7 to 12-year-old range. And this was a data set collected by Stuart Mostovsky at Kennedy Krieger Institute. And what Dina in our lab found was that there's 3 executive function classes, above average scores, and impaired. And not every child with a diagnosis falls cleanly into the impaired category. In fact, a good 18% of the average executive function group is kids who have autism. And there's a lot of heterogeneity there. So it's not that everyone with a disorder or diagnosis has this same kind of deficit. So Adriana Baez in our lab replicated this on our own data set of kids with autism and typically developing kids. Here, a big chunk of the average executive function group, 31% of kids with autism were basically unimpaired when it came to executive function abilities as reported by their parents. So this suggests, of course, that not everyone is going to be showing the same profile, and not everyone should get the same treatment, of course. So one of our hypotheses was to look at whether individual differences in brain dynamics or intrinsic connectivity of network dynamics, if that was associated with some of these differences in autism. So perhaps there's fewer transitions between brain states, and that may be one correlate of inflexibility. So Jason Nomi here looked at some adults with autism in the Autism Brain Imaging Data Exchange, which is many people have contributed data set to this data sets. And again, he did the dynamic sliding window approach and found evidence here for forced connectivity states. But the interesting finding was some evidence for reduced transitions between brain states in adults with autism compared to the typical adults. And actually, I think a few other groups have published similar findings now. So this looks like something that does appear to be the case in autism. We have other analytic approaches. For example, here's one called co-activation pattern analysis that instead of using a sliding window of a fixed window length, does a time point by time point co-activation pattern and groups together the states that are similar. But again, lets you calculate things like dwell time, transitions between states, and frequency of occurrence of brain states without the need for sliding windows. And Emily Marshall in our lab did an honors thesis where she focused on the cingulate and insular, the midcingulo-insular network, and asked, if the co-activation patterns with the rest of the brain differentiated kids with autism and typical kids. And here in 7 to 12-year-olds, she found evidence that a co-activation pattern that includes all of these regions, the salience and the regions here at the bottom, was observed less frequently in kids with autism compared to typically developing kids. So this one integrated brain state was not seen as often in the kids diagnosed. So a couple of folks in the lab, Lauren Kupis and Bryce Dirks, were interested in exploring this question, not just in the resting state, but in a task setting. And here they have a simple attention task, a simple flexibility task, where kids are asked to point to the object that's different. And sometimes the object differs on its shape and sometimes on color. And in a mixed block, shape and color can be intermixed among the trials. And that's more difficult than that requires flexibility. But of course, this is an easy enough task that all the kids can do it in the 7 to 12-year-old range. So what we're really interested in is how the brain dynamics that occur throughout this process. And we found these different co-activation patterns. I'm here focusing really only on the 3 main networks I mentioned earlier, the midcingulo-insular or salience, lateral frontoparietal or executive control, and medial frontoparietal or default mode. And there's different patterns of co-activation in which these networks either come together or come online in isolation. And what we found was that across different runs of the task, kids with autism do really well and don't differ from their typically developing peers. But towards the end, in the final run of the task, they actually engage more co-activation pattern 1, which is characterized by executive control control regions, they need to rely on that network more to get the task done as they move on through the task. So this suggests that dynamic functional connectivity approaches can be used to reveal atypical patterns of brain dynamics in prevalent neurodevelopmental disorders that are characterized in part by cognitive inflexibility. And the extent to which these brain dynamics underlie individual differences in flexible behaviors is something we're currently investigating. Now, this is all towards the goal, at least in the United States, we always talk about the research domain criteria or RDoC approach, which is trying to get us towards precision medicine and psychiatry. So according to DSM categories, we group things together by whether the patients are exhibiting the same symptoms, and this is done by clinician observation. But the idea of RDoC is to use integrated data across genetic risk and brain activity and physiology and other markers to try to arrive at data-driven categories or data-driven clusters of individuals who are more similar in a lot of ways to each other that can then be treated in ways that are more individualized. And the reason I show this slide from Insula and Cuthbert's 2015 paper is because they, for some reason, mentioned the insula in their diagram, and it could have just been an accident, or it could have just been coincidence, or it could have been because they've read all of our work and are convinced that the insula is the most important brain region to study. But in any case, I think we're very interested in the insula across these different conditions, because anyone who studies, anxiety or depression or schizophrenia or dementia has probably seen insular cortex implicated in their own data sets and their own findings. And so I think the insula in particular, with its role in brain dynamics more broadly and supporting flexible behaviors, is probably an important place to look for biomarkers across different conditions. But the specificity of the biomarkers is, of course, going to be the question. Now, this is something totally different that our lab stumbled upon that I would like to just end on. And that is that we collect all our data in South Florida, and many of our participants come from bilingual families just by chance. And we were never studying this phenomenon before, but I understand that there's some suggestion that there may be an executive function advantage in some individuals who are bilingual. And that hypothesis comes from the idea that switching between languages frequently relies on inhibition and other executive functions that might be broadly enhanced as a result of a lifetime of having this kind of bilingual switching experience. Now, many of the large-scale studies in adults and typically developing children have not found evidence supporting the bilingual executive function advantage hypothesis. But what has been shown is that in dementia, for example, there seems to be a offset or less cognitive decline in individuals who are bilingualism with age. And there does seem to be some suggestion that in children at the developmental side of things, that children coming from a low socioeconomic status tend to show this bilingualism advantage. So there does seem to be some maybe special cases or special instances where bilingualism might confer a particular advantage in this domain. And in the developmental, neurodevelopmental disorder and just developmental disabilities, the common wisdom for a long time was to not expose children to bilingual environments if they were experiencing language delay, because it might further delay their language production. But the more recent studies have suggested at least no negative effects to language development or to cognitive development in children with autism. There's been a lot of reviews on this. And in fact, there's some suggestion that bilingualism might even mitigate difficulties with flexibility. And more studies are coming out suggesting that there can be definitely benefits to promoting bilingualism in the home for those kids who come from such homes. So I think one of the most interesting findings is this that comes from Lauren Kenworthy's group, where parents seem to be reporting fewer executive function problems and repetitive behaviors in young dual language learners with autism. So there does seem to be an unexplored area here where there's potential clinical applications of just a simple natural intervention, which is tell parents to go ahead and be bilingual in their homes if that is the kind of home they're raising their child in. So Celia Romero in our lab is the one who first discovered this in our own data set. She noticed that we had a good proportion of our children were coming from bilingual homes. she had already collected the behavior rating inventory of executive function and just compared the data we'd collected so far in kids who were monolingual versus bilingual. And just as the literature suggests, in the typically developing kids, being monolingual or bilingual doesn't seem to affect the parents' report of executive function. But in the kids with autism, they're generally impaired overall on executive function, but the ones who are coming from the bilingual home seem to be a little bit protected or a little bit better in their executive function as reported by the parents. So this is some initial evidence that there might be an advantage here in kids with neurodevelopmental disorders, and we're trying to get a grant to study this right now. So hopefully we'll be able to do a large-scale study of this in the future. So most of the time we do all this work at the typical development level, across developmental disorders, to help us identify the neural circuits which we think are most important for flexibility broadly, and help us figure out the kinds of interventions that would target those circuits and probably lead to the best outcomes for those who have difficulties with flexible behaviors. And hopefully by the end of the day, get children to focus less on what they're wearing on their feet and more on their interactions in the world and on their daily lives. So this is the group that does all of the work I've showed you today. Of course, we can't meet in person. We haven't in a long time, but we're still working away virtually from different parts of the world where we are. And this is my collaboration network on the right, which I love to do work collaboratively and always like to add nodes and edges to this, this growing network. And these are the funding bodies and many collaborators who over the years have provided us with data sets, with methodological tools, and of course funding is always important too. But we just love working with other groups because I think the best science is done in this way. And I'm currently, of course, in San Diego on sabbatical, and really happy to have a chance to hopefully have some extended discussions with all of you. Thank you very much.
Thank you very much. You can open your mics if you want to, like me, clap for real. But if you clap virtually, that's okay too. I'm not, I don't think we're judgmental anymore in any true sense. Thanks very much, Lucina. Ultra clear. I didn't expect, anything less, but there were already quite a few questions. Okay. Sure. And Trevor already has a question. So why don't we start with Trevor, and then I read the others. And then more people will come.
Okay.
Hi, Lucina. Thanks for the talk. I just wanted to start us off with a rather conceptual question for you. So cognitive flexibility, right? To what extent is it really any different from cognitive control, executive function? Are we postulating something which is quite specific here, which in fact is not specific? To what extent does flexibility dissociate from, say, working memory, cognitive control? And is cognitive flexibility one thing? Is it unitary? I mean, perhaps there are several forms of flexibility. You can imagine flexible switching between inputs, outputs, and goals, and at all levels of the nervous system, including the motor output. So what is cognitive flexibility?
This is a lovely question, and the one that is very tricky, because when I first got into this, I started reading all the different accounts of executive function, and starting with Miyake and Friedman, and the unity and diversity of executive function, and as you are correctly pointing out, and as we see in meta-analyses all the time, all of these processes involve many of the same brain regions. So whether we're talking about inhibition or working memory or shifting, what people call flexibility. So we have all these cognitive constructs which map very closely, to each other on the brain, even though we like to dissociate them. So I may be agnostic to the question of whether this is different from cognitive control, whether we should call it something different, because as you see in the literature, people are seeing the similar brain regions across, all of these paradigms and across all of these terminologies. So I don't know if this is a question of semantics, if it's a question of tradition, or a real distinction between these different terms. What I do know is that all of these processes, whether you...
Yeah, but you did provide some evidence
Control,
that
cognitive control, flexibility.
just at the neural level, though, you seem to be implying that the singular opercular system may be included in the inferior frontal junction,
Sure.
which has some special arbitration role, which is quite distinct from frontoparietal network mediating classic cognitive control working memory. So I'm just wondering which side of the fence you're on.
Yeah, which side of the fence I'm on. I mean, I think that I think you're absolutely right. There's probably more than one domain or more than one way of thinking about flexibility, right? And so when we operationalize it in this very clean executive control kind of way, that's probably different than the some of the daily life examples, are different than things that involve emotion or that that are affect laden. So I think in terms of, I don't know if I want to fall on any side of any debate per se, but I do think that lateral frontoparietal and cingulo-insular networks are involved across all of these aspects of executive function, one of which we might pull out and call that one cognitive flexibility. If you look at, Miyake and Friedman's conceptualization and think that you can pull out this one concept. But because there's so much overlap, I think it's a little bit, personally, I think it's a little bit conflated by the different terminologies that people use. Like oftentimes you and I might be referring to the same phenomenon, but you've called it cognitive control, I've called it flexibility. And then when we look at each other's literatures, we might, I had to read like every paper under the sun in order to write these reviews because there were so many different terms to refer to what I thought was flexibility. So I don't think I even fall on any side of the debates at this point, other than to say, these are all the brain systems that are involved in all of these processes. And if we want to get any clearer on this, we do need to come to some semantic distinctions that I think will hopefully get us all on the same page.
Thank you.
Thanks.
Super. I have 3 questions, but Alex raised his hand. I guess he's going to ask his question that was online.
Hi, Lucinda. Thank you very much for the talk. Really, really interesting. I have to agree with Tristan that it was really clear. Plus, building upon what Trevor said about the uncertainty of cognitive domains, because I mean, I study a few different ones myself: working memory, executive function, sustained attention, and there seems to be a bit of a blurriness I found with them. But I'm really excited to see the insular and the inferior frontal areas and different areas of the cerebellum important in your research, because I look at traumatic brain injury, it gets a little more blurry, I have to say. But I was just wondering, I employ like a dynamic causal modeling, and I was wondering why you favored Granger causality as opposed to it, because I was told, oh yeah, I'm going to do Granger causality, it seems really good, and then I was told, no, don't do that.
That's another,
I was wondering if it was restricted by the number of ROIs you chose, or if it was a personal preference.
Yeah, I think it's just more happens to be, the lab I grew up in, but you can find the similar results to what I showed with DCM. I think a lot of labs have now used DCM to show, okay, you can find the insula, controlling these other regions. So I think in terms of methodological convergence, I've seen a lot of overlap between what you find in this particular set of nodes. I've seen a lot of overlap with the DCM and the Granger results. As you know, like DCM is more confirmatory and Granger is more exploratory. So it's a little bit, it's a little bit like the theoretical framework that you come from. And there's the pros and cons to Granger is just, at the time series level without making any assumptions about the underlying neuronal states. And DCM makes a lot of assumptions about the underlying neuronal states. So I think it's a little bit of a just tradition that you come from, but I was heartened to see a lot of overlap between the findings across these different causal modeling approaches, not from our lab, but just across the, different people doing it. So I'm happy to see it, at least some convergence there.
No, that's really good. I can come back to my supervisor then and say that. I think you were wrong. No.
No, you can try it. And I mean, my answer is always try it and see what happens. If they're really divergent, then we have a problem. So.
No, I did try them, and it was. I also felt validated that there was similar results when I looked at it. But yeah, thank you very much. Absolutely brilliant.
I'm feeling validated as well. That's always nice to hear. There's some convergence.
Yeah. Thank you.
I'm gonna ask Reza and Anujan to wait 2 seconds. And if you could read, if you can see, you could read the one from Sarah and the other one from Ekim, so before we go back to the hands.
Yeah, Sarab, I think, is presenting in our lab meeting in a few weeks. So thanks for joining. And so a bit methodological question. Are there any comparison metrics for dwell times transition states computed to those from hidden Markov modeling? So I haven't tried, are you talking about like Mark Woolrich's approach for dynamics?
Yes, indeed.
Yeah. So that's a great approach, and I haven't actually tried it yet. So the answer is I don't know. But I wish I had tried it, because it's a good question.
So just to follow up on that, so what's the window length that you use for your dynamic FC, and are they even with different window lengths?
Yeah, so for this, one of the studies I showed you, the reviewer, of course, asked this question. So we did it like with 3 different window lengths, I think 45 seconds, 30 seconds, and 60 seconds, or something to that effect. And this was with the Human Connectome Project data. So of course the TR is short, like 0.7. So we tried it with multiple window lengths and found kind of broadly similar results across. This is the supplementary materials is longer than the original paper, kind of. So, but yeah, that's why, because window length decision making is very tricky. We've moved a little bit more to the coactivation pattern analysis, which doesn't have any sliding window approach at all. So, but I think I like the idea of using the hidden Markov modeling. I just haven't gotten around to implementing that in the lab yet. I think that's a nice way to look at dynamics, but I have to get onto Mark Woolrich's code and figure that out. So hopefully in the future.
Lucina, there was a question from Ekim asking whether you recruit only diagnosed confirmed cases as opposed to the classic or from other labs of the AQ test.
Yes, that's a good question. We are taking mostly diagnosed, confirmed, and then we're reconfirming them again, doing another ADOS when they come to us. So right now, the studies, we're just comparing, very much confirmed cases.
Super. Let's go to Reza, Anujan first, then Reza. Sorry, Reza. He was first.
There's somebody in that. There's one more. Should I answer this one, Deandra?
Wait. They've been waiting with their hands up.
With their hands up. Oh, I see. Okay.
Go Anujan, then Reza.
Okay.
Thank you for that great talk, Dr. Uddin. When I think about the insula, I think about you, and I'm really glad for a lot of the amazing work you're doing. So my main question is, when we think about all these networks, there are embedded subcortically in this low dimensional space. And we know from DBS that for Parkinson's or tremor, you want to target the thalamus or the basal ganglia to modify Parkinsonian symptoms rather than the cortex. So when I think about your mid-insular switching from the VMPFC and the DLPFC, I just wonder, how does this mid-cingulo-insular network imprint embed itself subcortically, and how would you propose to modulate that with electrodes if you had a chance in a site-specific and temporally precise manner?
So this is a great question. So most of what I'm talking about, like the outflow from the dorsal anterior insula, seems to influence the lateral frontoparietal and other networks. But of course, the anterior insula is getting its input from the mid and posterior insula, which is getting its input from brainstem and subcortical regions, including like amygdala. And I think I have a diagram in my 2015 paper that shows like all the subcortical inputs, which I can send it to you. But basically, I think, they would be hard to target, but I think it's all coming, through the posterior insula up, and being integrated then in the anterior insula. So then in terms of targeting, it would be tricky. Like, I don't think the circuit is as straightforward as it would be in some of the disorders you mentioned, where exactly which subcortical region goes exactly to which cortical regions. Here we have a few relays. So I'm not even sure how the targeting would be most effective, to be honest. But yeah, I hadn't really thought about that. It's a great question. I mean, all of these subcortical to the cortex are really important to consider, but oftentimes we don't put them in the dynamic causal model or into the Granger causal model or into any of these things. I know some folks are starting to remedy that by putting the thalamus in and putting other regions into their models, but I think it's not as well worked out for this particular circuit as it is for some of the ones where you're actually able to do the deep brain stimulation. So I think it's an important question that I can't tell you offhand what the answer would be.
No worries. Sounds like an open question.
Yeah.
Thank you.
Yes, thank you.
Sorry, Reza, I made you wait again. Go ahead.
So very, very great talk. So one of the psychiatric diseases in which cognitive flexibility appears to be impaired is obsessive compulsive disorder, because people keep doing the same thing. So have you anyone else looked at cognitive flexibility in OCD patients?
Yeah, that's a good question. I know there's a little bit of literature on this. We haven't done it ourselves. And it does seem to be a lot of, oh, maybe Trevor looks like he has done it or has the answer. Oh, but you're muted.
Yeah, we have about 20 years worth of data on this. So this is a huge field. And I can direct you to the most recent paper with on rest in state with MICA by Mathilde Vaghi in biological psychiatry, showing that extra-dimensional set shifting is related to lower connectivity between areas 1247 and the body of the caudate nucleus. And this dissociates from other executive measures which seem to be associated with connectivity in dorsolateral prefrontal to other domains of striatum. So I think we do find rather selective deficits in cognitive flexibility. The interesting question is to what extent they are causal in OCD symptoms or whether they are a consequence of the OCD syndrome. And
Thanks very much.
Yeah. and they also have deficits in reversal learning, which is a different type of flexibility involving orbitofrontal cortex and is remediated by serotonergic medication. So we know rather more about flexibility in OCD than we probably do in autism.
Yeah, that's actually a great point, which as I was trying to figure out this, convergence between the animal literature and the human literature, the reversal learning paradigms, often involve reward. And of course, that's very different than the kind of set shifting and, so the reward aspect of it changes everything in my mind. It makes it a much more, about motivation and about, not just executive function. So that is, and that's why I think it's interesting to think about the contributions of orbitofrontal and caudate and all these to the whole flexibility discussion, because you see it so often, like you said, in OCD or in just in the animal models. Like those are the regions that pop up for reversal learning. So why is it so so dry in the human brain when we look at frontoparietal or other regions? And I think the reward aspect to me must be making a difference there.
Super. We can go now to the Andres question.
Is this one in the chat?
Yeah, and also there in the screen.
Oh, great.
Go for it, Ranger.
Yeah, impressive talk. It was really inspiring. I was just wondering, in terms of collecting data with ASD populations, if you found any differences between what has been reported behaviorally or from the parental side and what you're actually observing in the functional data.
Yeah, I mean, I think a lot of folks have noted this, that there is often discrepancy between parent report or everyday functioning and laboratory-based measures of executive function. In fact, they don't often converge. And some of the thoughts behind this are that in a structured environment, if you say, sit here and do this task, then kids maybe can perform just fine. But in the real world, when you tell them, go figure out a way to get to school, that's much more unstructured and open-ended and anxiety-provoking situation. So the real world flexible behaviors as reported by parents can be very much more affected or show more impairment than what level. And a lot of thoughts have gone into, should we collect more ecologically valid measures? Should we collect more standardized measures? Should we collect both? What does each one tell us? And I'm finding it quite convincing that we should collect more ecologically valid measures, because those are how, in the daily life, if you're not able to switch between, different choices, that's what we really want to get at. And I know there's something called the flexibility scale that a group has produced in the US. We've been using that more frequently because it's more targeted on flexibility per se and less on broad executive functions. Short answer is there are lots of discrepancies between these measures. So the question is, which one should we focus the most on? We can get a lot of clarity on the cognitive and neural aspects, if we use the standardized measures, but we can't necessarily translate to real life.
Yeah, I just wanted to add one last bit, because Trevor has brought up something really important. I'm particularly interested in studying ASD characteristics within genetically diagnosed intellectual disabilities. And one thing that we actually saw, that inflexibility as measured from autism perspective, it's actually mediated with high levels of anxiety and hyperactivity. So if you ever get the chance to explore these dynamic interactions, even at the cortical level, it would be amazing, really. So I hope there will be some interconnections between these two fields eventually. Thank you.
Yeah, thank you. I think this is important to point out, is that we always try to put categories against each other, autism or ADHD, but there's so much comorbidity between autism and ADHD, and a lot of kids with autism have anxiety. I think it's 30%. So, these clean categories just really don't exist. And so we're trying to do more of those, comorbid, just take all comers kind of analyses. And they become very, we require many subjects then to get a good sample. But this is what the US is pushing for in terms of the funding agencies, is to really consider comorbidity and not exclude comorbidity, because it's there, it's the norm. Thank you.
Thank you very much.
Lucina, I wrote 7 questions, but I'm going to ask you probably only a couple.
Okay.
I'll be nice and also give the chance to other people if they are still sort of holding on because they thought that they were shy about asking questions. I have a geeky question and a more conceptual one. The geeky one is, I guess, is relatively simple because you partially answer, which is when you're doing the k-mean clustering, and then this is a data-driven system, so you get these sort of networks separated by the k-mean system. In other work, like Athena de Mercy in the source of consciousness and other core work of k-means, there's this sort of network that it's the closest to the structural network, right? So you get these 4, 5, 6, clusters, and one is the one that it's the less active and the more that converges to the structural, COCO map, if it's in, or the DTI one. Is something that you can speak in your work? Is one of these networks sometimes look less than the classic, connectivity functional ones and more towards the structure?
That's a good question. We haven't explored that in the dynamic states, but I would be curious to see, if the frequency of occurrence of the ones that match the structural is higher or lower than the frequency, probably higher than the frequency of occurrence of the ones that map less closely. I have not explored this, so it's a great, really good question to explore.
Yeah, I'll take a look also back at the papers, because it just came to my brain. This week I was revising this sort of this idea of functional to neuroanatomy in these k-means, HMMs, etc.
I like this question because actually I find something which I don't talk about in most talks, but when we look at adults, you often find a high degree of convergence between the structural and functional networks. And, There's a lot of overlap and correlation. But when you look at kids, we don't find the correlation between structural and functional connectivity across that we look at. It's like as if one is preceding the other, but they're not correlated yet. But in adults, they seem to be strongly correlated. So, so there's probably developmental differences there too. Very interesting to explore.
Oh, that's very interesting. I've never thought of it.
Yeah, it's very, very strange.
And then the more conceptual one, it goes back into this idea, of the heterogeneous sample, right? So in your sample that you have, a sizable sample, when you start to play with the covariates, comorbidities, regressors, etc., does it feel that it's cleaning up into some of the questions, or it already the statistics become, noisier? How does it work? Because I've been asking this question to people who have large data sets. Some people take the approach of parsing the variance and using methods of parsing the variance with the functional connectivity matrices, while other people say I'm defining my functional separation, like the k-means, and then I'm asking my cognitive question. So it's sort of this computational statistical attitude. So I'm sort of interested from a theoretical point of view. And I know I can ask you this question because you can merge the things. Otherwise, it would be,
Yeah, this is depends, I guess, what you're interested in. So I'm putting in the chat here. One of my students did a study where it's not exactly what you're asking, but she kind of parsed the data in many different ways. So, the data set I mentioned where you had a typically developing group, an autism group, ADHD comorbid autism. So they're all just, they're groups defined by, DSM, but, they're all just children with different attributes, right? So she tried to parse them different ways. So one is like comparing autism to ADHD and TD. One is comparing with the comorbidity. One is just comparing them across their executive function profile. So just forget about what diagnostic group they're in, but what executive function group they're in. And then one, so she did kind of like cut up the data in different sort of ways. And it's funny because it was kind of a null result. Like she didn't actually find a lot of statistically significant differences. This is at the whole, ICA independent component, whole network level. But the point is like you can, and then she used the same data set then later on to try to parse the heterogeneity by using the individual connectome maps. So let me show you this. So she basically tried to do what you're suggesting, like take this one data set and cut it up 20 different ways. And here it is. This is the other one. But at the end of the day, like we don't actually have a good story yet. Like we couldn't find stable subgroups using the brain connectivity, and we couldn't find that they parse the heterogeneity. We couldn't find that, the diagnostic group was even very good at telling apart, anything useful about the brain. So I think we don't have, actually, we might not have big enough data sets yet, and we might not be looking at the right features of the data yet in order to really understand the subgroups and really understand what differentiates, the different phenotypes. So basically, these are the two things I put in the chat are works in progress, but they're published, but they were getting at this question in many different ways, and right now not having any satisfactory answers as of yet.
Yeah, no, thank you very much. And it also allows us to sort of think, attack the question from different perspectives, from
Right.
the point of view of the computations.
Right. It's easy to do computations on any data set. The question is like, what's the appropriate way to think about the heterogeneity of the sample? What's the most useful clinically, or what's the most useful for producing neuroscientific insights? I think now we have so many methods and so many experts on helping us apply them. So really, like the hard part is coming up with the right question.
Yeah, I would imagine that we should be better at that than those who develop methods, hopefully.
Well, we should at least put on our philosophy hats and think about what would be the most useful.
Super. Thanks very much. I'll open again the floor for any questions, comments before we wrap up, and we let Lucina continue with her normal day, and we all go to our appropriate proseccos or whatever it's your a weapon of choice in this respect. I can reach the sword if someone wants a 1, 2, 3. No, good. So join me in thanking Lucina again for a great talk and a good discussion.
Thank you so much. I hope to see you all in person sometime in the future.
We hope to see you around in Europe doing a cool tour.
A tour.
Yeah.
I'll let you know. And thanks so much for the great questions.
Super. Thank you very much.
Bye. Bye.